Hello guys and welcome back to the channel. I am Tommy and today we're going to continue the microbiology series, the foodborne microbiology. In this video we're going to view in detail the control of the microbial growth in food. And before we start, make sure to hit the subscribe button below and comment I subscribe. It costs nothing for you, but it means a lot to me. Okay, thank you. Let's begin now. When we talk about the control of the microbial growth in food, we first have to know which microorganisms are present in food. So here we have them classified by types. We have the beneficial, the spoilage, the pathogenic, and the indicators. So these four groups are for all the microorganisms we're going to view. The beneficial are the fermented ones, the spoilage cause the decomposition of the food, the pathogenic cause diseases to the hosts, and of course infection and poisoning. And the indicators are, for example, the mesophiles, enteric such as Escherichia coli, the coliforms, the enterococci, even yeasts and molds, and the Staphylococcus aureus. This is very important, for example, for contamination via the nose, because Staphylococcus aureus lives in the nasopharyngeus. Food interest groups. The bacteria can be lactic bacteria, and lactic bacteria can be beneficial in some cases. Spoilage, they can alterate the food, and the indicators as well. The bacteria coliforms can be indicator and pathogenic. The acidic can be beneficial only. The butyric can be spoilage and pathogenic. And the putrefaction bacteria, spoilage. So as you can see, we have like the lactic that can be almost everything but not pathogenic. The coliforms that are like the bad guy because they can be pathogenic, and but they can also help us because they are indicators. The acidic, which are the best ones because they cannot be spoilage, pathogenic, or indicators, they are only beneficial. And the butyric are like the worst because they are spoilage and pathogenic as well. And then the putrefaction, it's not their fault, they basically spoilage. Then moving on, we have the yeasts, which can be beneficial, spoilage, and indicators. The molds. Molds can be everything. It can be beneficial, spoilage, pathogenic, and indicators. And then viruses and prions, and guess what? Pathogens, and they can be pathogens for human, but also for bacteria. The origin of the contamination, well, from the water, the soil, the air, and or the dust. From microorganisms naturally present in the food. From the manufacturing, the storage, and the transport, so how we actually create the product, how we create that food, and even in the supermarket and maybe at home, we can get the contamination. Factors affecting microbial growth in food are, well, the duplication of the bacteria is using binary fission. And we need to talk about growth as the population increase, not as the growth of an individual. Microbial growth curve, this is like, if we put it on a graph, the curve of growth will be dormant, so like this, dormant, then exponential will raise, stationary will stay, and then death, and will start to fall down as it starts to die, the population starts to die. And then the types of factors we have for actually controlling the microbial growth in food are the intrinsic factors and the extrinsic factors. The intrinsic factors are like food composition. If the food has low vitamin B, this all the group B of vitamins. If you want to know more about vitamins, check my video on vitamins in the nutrient description series. And well, if we have low vitamins B, well, the vitamin B dependent bacteria will not grow, and the vitamin B dependent, non-bacteria, I mean the bacteria that doesn't depend on vitamin B will grow. If there are high carbohydrates, well, the molds will probably start to grow faster, and if the food has a lot of fats and proteins, we will get putrefaction for the proteins and rancidity for the fats. Depending on the pH, the bacteria likes 4.5 to 9 pH. The molds can grow between 12 and 11, and the yeast from 2 to 9. Water activity, if we have high water activity, well, chances are we will have high microbial growth as well. And this is the oxidative reduction potential, and indicates the amount of air. And of course, if there are high amounts of air, high amounts of oxygen, well, the aerophils will start to grow, the aerobes bacteria will start to grow, and the anaerobes will not, and the reverse happens. The physical structure of the food, yes, because if we cut the food, we will actually destroy the natural protection and increase the contact surface. So having more surface, the bacteria can actually affect it more efficiently. Presence of natural antimicrobial substances, yes, because some food has some antimicrobial substances like coumarins, lysozyme, Tabasco, the polyphenols, the allicin and the catechol, the acetic acid, the lactic acid, and the bacteriocins. Moving on, we have extrinsic factors such as temperature. Well, if we have high temperatures, probably high growth, same as humidity. And then in the storage atmosphere, we talk about percentage of oxygen. As I said before, this is like around the food, this is inside the food, the amount of air inside the food, this is around the food. And yes, if there is high amount of oxygen, well, the aerobic growth will start to increase, and the reverse happens. If there is low amount of oxygen, the anaerobic bacteria will start to grow. And then if there is high CO2 in the air, well, microbial growth will be lower. Control of microbial growth in food. How we do that? Well, we have to work with populations, not individuals, as I said. And here we have the concept of decimal reduction. That's the 90% less of the population. That's a decimal reduction. So, having that in mind, we can say that cleaning is the separation of microorganisms, and it has only one decimal reduction. But if we use water at 30 degrees Celsius, we will get from 4 to 5 decimal reductions. Disinfection is the destruction of pathogens, D8 from D8 to D13. And then sterilization is the destruction of all microorganisms. And well, this is an error because in here actually we have no microorganisms. So it's not like a decimal reduction, we have no microorganisms. Preservation, we get preservation after using these methods to ensure the quality and the safety of the product. And we have to classify the agents as microbiostatic, that inactivates the bacteria, or microbicidal, that kills the bacteria. And the efficacy of these treatments depend on the size of the food, the intensity we will apply them, the exposure of the treatment, and the environmental conditions as well. And then we have the physical methods and the chemical methods. Physical first. Well, we have the filtration with depth or membrane filtration, high temperatures such as UHT, ultra high temperature sterilization and pasteurization, low temperatures such as refrigeration or freezing, and to lower the water activity, dehydration or addition of solids, the modified atmosphere with less oxygen and more CO2, as we saw, it lowers the microbial growth, and radiation as well. And then an interesting one, which is new, the high pressures. High pressures at 400 millipascal actually is 1 decimal reduction. On the other hand, we have the chemical methods such as smoking, and smoking create acids, aldehydes and phenols, but it has also the inconvenience of creating some cancerous substances. That's why we have to be careful with smoking. Then we have nitrates and nitrates, the sulfites, bisulfites. These break the disulfide bridges and degrade the sugars. If there are no sugars, chances are bacteria will not grow. Saturated fatty acids and the salts of calcium, sodium and potassium. The sorbic acid that inhibits the molds, the benzoic acid that inhibits molds and yeasts, the parabens, which are not pH dependent and they do the same thing as benzoic acid, similar, the organic acids that acidify the cytoplasm and denaturate the proteins, again acidity and has to do with pH, and bacteria don't like the pH too low. And then the phenolic antioxidants that act on the membrane, and the antibiotics, we all know that one. They have a side effect, which is the damage to the microbiota, the health microbiota, that one we actually need in our gut. And then the bacteriocins, which are dipeptides with antimicrobial function, and they added to the foodstuffs. Okay, so that's it for the control of the microorganisms in food, control the growth of them in food. And I hope you like this video. Click the like button below, share this video to friends and family, let them know, and see you on the next one.